In this activity, we're moving inside the plant and looking at the internal structure of a woody stem. You'll be working with a prepared cross-section of Tilia or basswood and learning how a stem that started out herbaceous becomes woody through secondary growth. Woody stems don't just start out woody, they begin as herbaceous stems. The original tissue layout sets up the framework for everything that comes later. In the center is the pith, and around it are vascular bundles that contain primary xylem and primary phloem. As the stem matures, a vascular cambium forms between the primary xylem and primary phloem and eventually becomes a continuous ring. This cambium is responsible for secondary growth. It produces secondary xylem inward, which becomes wood, and secondary phloem outward. When you're looking at the secondary xylem, you're not just seeing one uniform block of tissue, you're actually seeing layers that reflect how the tree grew over time. During the spring, when conditions are usually favorable and growth is rapid, the vascular cambium produces what we call springwood, also known as early wood. Springwood is characterized by larger vessel elements and thinner cell walls. These wide vessels are great for moving large amounts of water quickly, which makes sense because the tree is producing new leaves and growing rapidly at this time of year. As the growing season continues into summer, and conditions often become drier or growth slows, the cambium starts producing summerwood or latewood. Summerwood has smaller vessel elements and generally thicker cell walls. This tissue is stronger and denser, and while it doesn't move water as quickly, it provides more structural support. Together, one band of springwood followed by one band of summerwood makes up one annual ring. So when you see alternating light and dark bands in the wood, you're really seeing a record of fast growth versus slower growth over the course of a year. As you examine your specimen, try to notice how the springwood zones look different from the summerwood zones, not just in color or thickness, but in what they tell you about how the plant was growing at different times of the year. When we talk about using growth rings to study trees, we're really talking about a field called Dendrochronology. Dendrochronology is the science of dating and studying trees using their annual growth rings. Each year, a woody plant forms one growth ring made of springwood and summerwood. The width and appearance of that ring depend on how good the growing conditions were that year. In years with plenty of water and favorable temperatures, the rings tend to be wider because the tree grew more. In years with drought, cold, or other stress, the rings are usually narrower because growth was slower. Scientists don't just count rings to figure out how old a tree is, although that's one important use. They also compare ring patterns from many trees in the same region, because climate affects many trees in similar ways. Trees growing in the same area often show matching patterns of wide and narrow rings. By lining up those patterns, researchers can build long timelines that go back hundreds or even thousands of years. This lets scientists use trees as natural climate records. From tree rings, they can infer things like past droughts, wet periods, fires, or unusually good growing seasons. In other words, when you look at a woody stem under the microscope and see annual rings, you're not just seeing plant anatomy, you're seeing a biological record of environmental history written into the wood. So dendrochronology connects plant structure, plant growth, and Earth's climate history in a really powerful way. When you look at the secondary xylem in a woody stem, you're not just looking at one uniform tissue. It's actually divided into two main regions, sapwood and heartwood. Sapwood is the younger outer part of the xylem. This is a region that is still actively involved in water and mineral transport from the roots up to the leaves. The vessel elements and other conducting cells in the sapwood are functional, and this is where most of the day-to-day movement of water through the tree is happening. Sapwood also often plays a role in storage of substances like starch and other nutrients. As the stem continues to grow year after year, older xylem in the center of the stem gradually stops functioning in transport. This older xylem becomes heartwood. Heartwood does not conduct water, but it is still very important. Its main role is structural support. It helps keep the tree upright and strong. Heartwood cells are often filled with resins, tannins, or other compounds which make the heartwood darker in color and more resistant to decay, insects, and pathogens. That's why heartwood is usually the part of the tree that's valued for lumber. It tends to be stronger and more durable. Xylem and phloem rays are bands of mostly parenchyma cells that run radially across a woody stem, from the center toward the bark. While xylem and phloem mainly move minerals up and down the plant, rays allow lateral or side-to-side transport. Xylem rays help move water, minerals, and stored nutrients across the wood, while phloem rays help move sugars and other organic materials across the phloem region. Because ray cells are usually living, they also play a role in storage and internal communication between tissues. When you see light-colored or distinct bands running across the stem in cross-section, you're often looking at these rays connecting the plant's transport systems. Outside the vascular cambium is the secondary phloem, which is responsible for transporting sugars and other organic compounds produced by the leaves. This tissue also contains phloem rays, which help move materials laterally across the stem. Beyond the secondary phloem is the periderm, which replaces the epidermis in woody stems and is made up of cork cambium, and phylloderm. The cortex lies between the periderm and the secondary phloem, although in older stems it may be compressed or partly lost. All of the tissues outside the vascular cambium together are called bark, so bark is not a single tissue but a combination of several layers with different functions. As you think about this, consider two perspectives. First, what do annual rings tell you about how the plant grows and functions over time? Second, why might those same rings be useful to scientists studying age or environmental history? As you think about this question, consider what annual rings represent in terms of how the plant grows over time. Think about how changes in growth rate during different seasons create visible patterns in the wood. Then think about how those same patterns might be useful to scientists who want to learn something about a tree's age or the conditions it grew under. Try to connect plant function with how people can use these rings as a source of information. Write your prediction before you start observing. Before you start drawing, let's do a guided walkthrough of what you should be able to find in this woody stem cross-section. Start at the very center of the stem. The tissue right in the middle is the pith. In some specimens, it's solid, and in others, it may be partially broken down or hollow, but it always marks the center of the stem. Moving outward from the pith, you'll enter the large region of secondary xylem. This is the wood. This is usually the widest part of the cross-section and takes up most of the stem's area. Are you able to count the annual rings of the 4-year-old stem? First year, second year, third year, and into the 4th year. Within the secondary xylem, look for large, round, or oval openings. These are vessel elements, which are part of the water conducting system. You can see the springwood where there was a lot of large vessels, while in the summerwood there are smaller vessels. Also within the xylem, look for dark bands or lines that run radially from the center toward the outside. These are xylem rays made of parenchyma cells that move material sideways across the stem. Now move outward to the thin ring between the xylem and the phloem. This narrow layer is the vascular cambium. It's usually hard to see as a thick structure, but its position right between the xylem and phloem is a key clue. It is stained green in the older stem. Just outside the vascular cambium is the secondary phloem. This region is much thinner than the xylem in most woody stems. Within the phloem, you may also see bands of cells running radially. These are the phloem rays, which are similar in function to xylem rays, but located in the phloem tissue. Moving farther outward, you'll reach the protective outer tissues. The outer covering is the periderm, which includes cork. This replaces the epidermis in woody stems and forms the protective surface. Between the periderm and the secondary phloem is the cortex. This is much thicker in the 1-year-old stem and gets thinner and disappears altogether in older stems. Finally, remember that bark is not just one tissue. Bark includes everything outside the vascular cambium, which means it includes the secondary phloem, cortex if present, and the periderm. Take a moment to visually locate each of these before you start drawing. The better you orient yourself now, the more accurate and confident your labeled diagram will be. Now it is time to draw the 4-year-old basswood stem. You don't have to draw the entire thing, only a quarter of it. Your drawing should include the pith, secondary xylem and phloem, vascular cambium, xylem and phloem rays, vessel elements, periderm, cortex, and bark. Use their relative positions to help you. What's in the center, what forms a ring, and what lies toward the outside of the stem. Pause the video to make your drawing. Answer these compare and contrast questions. How does the amount of space occupied by secondary phloem compare to that of secondary xylem in the specimen? Think about proportion and function. What tissue makes up most of the stem, and why might that make sense for support and transport? Xylem rays and phloem rays both function in lateral transport of fluids. In what ways are their structures similar, and how do they differ? Focus on what these rays do and where they are located. Both move materials sideways, but one is in xylem and one is in phloem. Ask yourself how that difference in location might affect what they look like or what they carry. Take a moment to pause the video and answer the questions. This activity shows you how secondary growth actually builds the woody structure of a stem over time. It connects microscopic anatomy to things you can see in real trees, like growth rings, wood, and bark, and once again reinforces the idea that plant structure is tightly linked to function.